AP Bio. So we're going to take a look at topic 6, Group A questions. Again, these deal with DNA, RNA, gene expression. And again, remember that you're going to be given questions that focus on an experiment based in the context of different topics. And so this is a classic example of that. This is actually off of one of the newest AP tests, newest AP exams. And again, this is focusing on looking at RNA polymerase and what happens with these RNA molecules if you inhibit them. And so let's take a look at this. So we had scientists studying transcription in yeast, Saccharomyces cerevisiae, S. cerevisiae, and they created an experimental strain that produced a modified RNA polymerase that had a single amino acid substitution in it, right? And so they modified the RNA polymerase, right, the enzyme. And then the scientists determined that the maximum elongation rate during transcription with and without the modified enzyme can be shown below, okay? And so what they did is they showed you the maximum rate of the wild strain and the experimental strain under natural conditions, and this is what happened when you added that amanitin, right? And so figures that the solution we know is very toxic to mushrooms, and it's a specific RNA polymerase inhibitor. So if it's inhibiting the RNA polymerase, you can just imagine that it's preventing it from doing its job, right? And what it does is it binds to the active site. So in this case, it's binding to the active site and it's inhibiting transcription, right? So in a second experiment, they treated the wild type and experimental strains with the same concentration, okay, and they recorded the maximum elongation rate. You can see that under natural conditions, this is what it looked like, and then when you added that chemical, you wound up getting a really low rate. Just from looking at that, you can tell, okay? So they want 3 structural components of an RNA nucleotide monomer and explain the role of the RNA polymerase during transcription, okay? So what you needed to do for this was talk about what makes up RNA, okay? And we know an RNA nucleotide has the following components, okay? A ribose, a phosphate group, and a nitrogenous base. You got a point for that. What's the role of polymerase during transcription? Any one of these is fine. That it either synthesizes RNA molecules based on a DNA template, or what it does is it joins or bonds the newly paired RNA nucleotide and the growing RNA strand with a covalent bond, right? So what that means is that it's adding a nucleotide to the growing strand, right? Happens to be a phosphodiester bond, okay? But anything that relates to it creates new RNA, it uses the DNA template to create a new RNA molecule, that's all good, okay? Now, the dependent variable. Dependent variable is what you measure, okay? So in this experiment, they were measuring the elongation rate of mRNA. Remember, your dependent variable is always on your y-axis, okay? It's always on your vertical axis. So we're measuring the effect of whatever we're doing on the maximum elongation rate, okay? And we're comparing 2 strains. So we're looking at 2 different strains and how quickly they can elongate or make an RNA molecule, okay? Now, they want to know a control, right? Identify a control group missing from the second experiment, right? So they have the wild and experimental strain, right? So they want to know what's a control that's missing. So there's some possibilities here, okay? An appropriate control group missing from the second experiment. Okay, either of the strains from the first experiment, but without the amanitin, is an appropriate control for the second experiment. But there may be other appropriate controls. So what they should have done in that experiment is had either strain, okay, without it as a control to show that there would be no impact without it, okay? And the need for the control, because this shows the fact that if either the wild strain or experimental strain is selected, the justification is that the effect of amanitin on the maximum elongation rate cannot be determined without comparing it to the maximum rate under the same conditions without. Okay? So I'll give you a second to read that. I don't want to just dictate. Okay. And so let's take a look at the third component to this. Describe the effect of amanitin on the maximum elongation rate for wild type and modified RNA polymerases, and determine the ratio of the average maximum rate compared to the wild ones. So if you look at this, you can clearly see that there's definitely an impact, right? It reduces it. Okay, it makes it lower. Now to figure out the ratio, okay, so the ratio of the average rate in the modified RNA polymerase compared to the wild strain, okay, under natural conditions, we can see right here, it's 12 to 2, right, or 6 to 1, okay? And so you just simply do a little ratio, 12 to 2 or 6 to 1, okay? And that works, okay? And that's where you get your points. And I'll show you the answer for that one, okay? Okay. Moving on to the last one, the null hypothesis. Now, this is where a lot of people get confused. A null hypothesis is a hypothesis that indicates that there's no relationship between the variables being tested, okay? So that means that whatever you're testing, the null hypothesis would state that there's no impact. So there's no effect, okay? And the reasoning to justify the claim that the change in the sequence affects the shape of the active site. Okay, so let's take a look at the answers on that. So first of all, your null hypothesis would be that the modified polymerase does not affect the maximum elongation rate, and any difference between the two rates is due to chance. And the reasoning for this is a change to the active site would explain the decrease in the elongation rate. Okay, and we see that. Okay, by changing the active site, by having that one modification there, that would explain the reason why there's a decrease in the rate of elongation. Okay, all right, so that's it for the first question. 10 points total. Okay, you got a point for each part of D here where you have to state it and then justify. Okay, you got 2 points for part C. Most of your points were in part B. Okay, where you had to discuss the variable, the control group, justify the reasoning for the control group. Until we gave 2 points for this. Okay, or they gave 2 points for this. Okay, moving on to the next one. You have this amino acid sequence, and each amino acid is represented by a 3-letter abbreviation. So they said, assuming that species 1 is the ancestral species of all the others, okay, explain the genetic change that produced the polypeptide we see in species 2 and the change in 3. So what we saw was a mutation, right? In this case, it was a substitution. We went from val to lys, okay, lysine. And in this case, it's a deletion, okay? So we saw a deletion, okay? So for 1 to 2, it was a substitution. From 1 to 3, it was a deletion, okay? And the result of the change on the polypeptide would be, by doing this, you're changing the amino acid sequence. So that changes the sequence in the polypeptide, okay? In this case, what you're doing is you're terminating the polypeptide after the valine at position 8. Okay, there's nothing else beyond that. So your protein is shortened, right? This here is going to create a truncated protein because you are missing amino acids, so your protein is incomplete, okay? Then they said, explain the effects of the mutation on the structure and function resulting in the protein in species 4. Okay, so they want to know, okay, here's species 4, and look what happens here. We have a whole bunch of substitutions here, okay? So what could happen? 3 possibilities. Okay, the first possibility is the protein has a different structure and a complete change in function, right? And that's because we're changing all those amino acids, and that's going to result in a complete shift in the sequence, and it'll interfere with the structure, which changes the function. Okay, the second thing is it may have a different structure, but the function is going to be the same. And the reason for this is this might be affecting another part of the protein. Doesn't affect the active site, doesn't affect the regulatory site. So this could be a mutation in a part of the protein that doesn't do anything and doesn't have any direct impact on its function, okay? And then the last one here, this is a good one. This is the cop-out one. There's no change in the function or structure at all because whatever you've done, you've changed the amino acid sequence, and it doesn't affect it. So you can change sequences, and this can happen. Maybe these amino acids happen to have the same chemical characteristics and chemical properties as the original sequence. And so as a result, the protein is thankfully unchanged, and you can still carry out whatever function needs to happen with the protein. Okay? And that's very possible. It's a realistic answer, and actually, if we had covered evolution, we would find out that you can have many changes in the amino acid sequence of specific proteins, and there's no loss of function. They still work. Okay? So that's it for this part. Again, check your answers and keep plugging away, guys. We're at the end. This is it. We've made it. Just a couple more days of work, and we'll be ready to rock for this exam. Okay? Stay safe, be well. We'll talk to you soon. Bye.